and welcome to some biology class. Friends, today we shall discuss about apoplastic phloem loading. In apoplastic phloem loading, during the transport of sugars from source cell to the sieve tube element, at some point the sugars enter into the apoplast, from where the sugars are transported into the sieve tube companion cell complex by a H plus sucrose symporter. This type of phloem loading is seen in families that are regarded as highly evolved. This is more efficient than symplastic phloem loading because it achieves higher rate. It is insensitive to low temperature, thereby this type of loading pathway is seen in plants that grow in temperate zone. In apoplastic pathway, a H plus sucrose importer is thought to mediate the transport of sugars from the apoplast to the sieve tube companion cell complex. It is a secondary active transport that is energy dependent, and the energy comes from a proton gradient which is established due to the pumping of protons by H plus pumping ATPase. This figure explains the ATP dependent sucrose transport in sieve element loading. This is the cell membrane of the companion cell. This is the protein pumping ATPase, and this is the sucrose H plus importer. The function of proton pumping ATPase is to pump proton from inside of the companion cell to the outside. For this pumping, energy comes from the hydrolysis of ATP. Due to the pumping of protons from inside of the companion cell to the outside, the concentration of H plus ion increase in the apoplast, whereas the concentration decrease inside companion cell. So this difference in H plus concentration constitute the proton gradient. This proton gradient provides energy for the symport of sucrose and H plus through the plasma membrane by a sucrose H plus symporter. So in this process, the necessary energy comes from the proton gradient, and the proton gradient is generated by proton pumping ATPase, which is present in the cell membrane of the companion cell. In many plants, intermediary type of companion cells are absent. In such plants, ordinary type of companion cells and transfer cells are present. Transfer cells are unique cells having wall ingrowth. The ordinary type of companion cell or transfer cells have very few plasmodesmatal connection with the surrounding mesophyll cells. So, this type of companion cells are symplastically isolated from the surrounding mesophyll cell, whereas the companion cell has numerous plasmodesmatal connection with the sieve tube. Because such type of companion cells are symplastically isolated from the surrounding mesophyll cell, because there is no plasmodesmatal connection, so symplastic phloem loading is not possible. In such plants, apoplastic phloem loading occurs. In apoplastic phloem loading, sugar enter into the apoplast at some point during their transport from mesophyll cells to the sieve tube companion cell complex. This figure explains the process of apoplastic phloem loading. This is the mesophyll cell where sucrose molecules are synthesized. And this is the companion cell. It is a transfer cell having wall ingrowth. In between mesophyll cell and the companion cell, there is cell wall, and the cell wall is the apoplast. Between the mesophyll cell and the companion cell, there is no plasmodesmatal connection. So the companion cell is symplastically isolated from the mesophyll cell, whereas the companion cell had numerous plasmodesmatal connections with the seed tube element. So in apoplastic phloem loading, sucrose molecules are discharged from mesophyll cell into the apoplast, possibly by the help of carriers. From apoplast, the sucrose molecules are actively transported into the companion cell by H+ sucrose importer, which is present in the cell membrane of the companion cell. It is also present in the cell membrane of seed tube element. So, with the help of H+ sucrose importer, sucrose molecules are actively transported from apoplast into the companion cell. The secondary active transport is driven by proton motive force. The proton motive force means the proton gradient, which is established with the help of a H+ adipase. From the companion cells, sucrose molecules can be diffused into the seed tube element through plasmodesmata. It may also be transported from companion cell to the apoplast, from apoplast to the seed tube through H+ sucrose importer. And in this diagram also, you can see that from this phloem parenchyma, sucrose molecules are discharged into the apoplast, possibly by means of carrier, and from the apoplast, the sucrose molecules are actively transported into the companion cell by means of H+ sucrose importer, or it can be actively transported directly from apoplast into the seed tube. So friends, this is all about apoplastic phloem loading. For more update, please subscribe some biology class, and also please share your comments.